Good morning, everyone, and thank you for being here, giving me the opportunity to introduce you to Caladrius and to talk a little bit about our programs and what differentiates us in this very exciting field of cell therapy. Like all the public companies, I'll just remind you, I'll be making some forward-looking statements, and I'll ask you to please keep those in mind as you make any decisions about our company going forward. If I start with a simple overview of Caladrius, like most cell therapy companies, we have a very exciting and we believe dynamic pipeline focused on the development of therapeutics and therapies. But what makes us unique is that we combine that pipeline with one of the premier cell therapy service providers in the areas of process development, process optimization, and manufacturing that exists in the industry today. And we believe that combination gives us not only a competitive edge as it relates to the efficiency and effectiveness of the development of our own programs, but allows us to provide to our clients a similar level of efficiency and effectiveness. Our lead clinical program is in the area of immuno-oncology. It's a program that is in phase 3 today in metastatic melanoma, and it's been granted fast track and orphan drug designations as well as a number of other endorsements, which I'll speak to later. We have 2 additional platforms that we're developing, one in the area of ischemic repair and the other in the area of autoimmune disease, and I'll speak to those very briefly. And I'll mention that we have now, since January, with the variety of changes that I've made on the management team, a very strong and experienced management cadre that is complementary now to the very strong scientific team that has been the hallmark of Caladrius in the past. So one of the things that sets us apart from many other companies is the fact that all of our development programs share a common root. I like to use this analogy because I often spend time in the Rockies skiing, and when you're in the Rockies and you see these groves of aspen trees, you think, wow, those are lots of trees. But I've learned botanically that they're actually single organisms. They share all these common roots. And that's really how you should view our pipeline. Even though we're in oncology, ischemic repair, autoimmune disease, it's really a similar root technology. It's an autologous approach to cell therapy, using cells from a patient to prepare a therapy that's specific for that patient. On the other side of the coin, I want to take a moment to just talk about PCT. It is a single source premier cell therapy service provider, and what we're able to do, not only for ourselves but for our clients, is provide a complete solution on the GMP manufacturing, process development, and regulatory development of cell therapy products. We're capable of providing process development, process optimization, automation, scale-up, clinical and commercial manufacturing in a fashion that I think provides for our clients a cost-effective way to manage cost of goods across the spectrum of development from early stage to late stage, and allows those clients, and of course we're among them, to apply a philosophy of right timing to the next step in development, so investments coincide with advances in development and you don't have to spend all your money up front. We're very proud of our client list. We've got a number of people here in this audience and others that are recognized. This is just a select few clients. We've been in business for more than 16 years. We've had more than 120 clients, produced more than 20,000 individual products, and have worked with just about every cell type that has ever been investigated in cell therapy. So it's within a single group really have amassed a great deal of experience and expertise. So I go back now to the pipeline and introduce you in a little bit more detail to what we're doing. Let's start with our immuno-oncology program, which is CLBS 20. It's a program that's focused on stage 3 and stage 4 metastatic melanoma. As I mentioned previously, it's been granted all of the regulatory and external scientific endorsements that one might expect from a very promising new therapy. It has fast track and orphan drug designation, as well as a special protocol assessment for the phase 3 program from FDA. It's also been granted Advanced Therapeutic Medicinal Product classification from the EMA, and we're very proud to say that we are the recipient of approximately $18 million from CIRM just last spring in support of this program, and more recently another approximately $2.3 million from NCI in support of this program. Why are we working on melanoma when so many people look at that and say, wow, that's a really crowded space, right? Well, we're working in melanoma for two principal reasons. One, actually it's three reasons. One is there's still a major unmet medical need, notwithstanding the great advancements that have been made recently with the checkpoint inhibitors. Second, it's one of the least homogeneous of all tumor types, and it's a place where intuitively one might expect an autologous approach to yield the greatest results. And three, it's an area where everyone else seems to be working on one side of the challenge or the paradigm, looking at how to improve an already existent immune response. We're trying to provide an immune response, and we're doing that through a technology that takes use of progenitor cells from the tumor and dendritic cells from that same patient. We prepare our cancer vaccine in a very simple way. It makes it a cost-effective approach and one that is consistent with current standard of care. We begin with the surgical excision of a skin lesion, a tumor, from which we can isolate the cancer-inducing stem cells or the progenitor cells. Those are expanded and then ultimately irradiated so they're no longer viable. And that becomes the source of the antigenic fingerprint for the dendritic cells and ultimately for that person's immune system. We are not isolating or trying to identify 1, 2, or 3 specific proteinaceous markers. We're actually using a holistic approach, looking at the complete antigenic fingerprint that is unique to that patient, and that really becomes the source for the patient's vaccine. We are, as I mentioned, in phase 3. I haven't brought the data slides here simply because we're running short on time, but we've completed 2 phase 2 programs here, both of which demonstrated truly compelling results, the ability to increase overall survival substantially, and that's an important aspect of our program. We're really focused on attacking the circulating cells that provoke metastases and ultimately cause the death of the patient. And so in our studies, overall survival is really the key endpoint. It takes a little longer to get there, but we've been demonstrating multiples of improvement on overall survival in comparison to standard of care and even in comparison to the combination products that have been recently reported. Our phase 3 program is underway. We're dosing patients, and we expect to reach our interim analysis after 99 events in the trial sometime in late 2017, and hopefully that will lead to a BLA in 2018. What's also exciting about this particular application is that it can be applied across a variety of solid tumor types, and we have programs that are planned and ready to initiate as soon as we identify some partnerships in the areas of ovarian cancer, hepatocellular carcinoma, renal cell carcinoma, and a number of other very important cell types. Our second program that I'd like to talk about is in immune modulation. This is actually a T-cell program that's focused on the treatment of early onset type 1 diabetes. And this is a strategic collaboration that we recently announced with the Sanford Research Foundation. We're very excited to be working with Sanford. They will not only provide clinical resources, hospital sites, and access to patients, but they also will be providing direct support to the trial. In autoimmune disease, and especially in type 1 diabetes, the simple explanation is that the body's own immune system, typically their T effector cells, are in fact attacking viable cells within the body, and in the case of diabetes, they're attacking the beta cells in the pancreas, which are those that provoke the production of insulin. If we can catch patients early enough in their diagnosis when there are still viable beta cells and protect those remaining beta cells, we should be in a position to prevent further progression of the disease and maybe even perhaps regeneration of those cells in the pancreas. And we do that with a T-cell approach that is actually very straightforward. In this particular case, we isolate T cells from a patient, we expand them, and then we return them to that patient in order to restore the balance between their T regulator cells and T effector cells, and that's expected to have a therapeutic effect. In fact, we've done 2 phase 1 studies with this technology. In one that was done in adults, we demonstrated the ability to actually manufacture the cells and return them to the patients, showed that the patients tolerated them as their own cells, and importantly, after radiolabeling them, showed that the cells existed in the peripheral blood for at least a year. So that means that there's a possibility of a durable effect. In a second trial done in Europe, in children, we demonstrate over the course of a year a remarkable reduction in the progression of the disease using standard markers for diabetes, as well as the ability to reduce significantly the amount of insulin that these early onset diabetics needed, and in fact, in certain cases, actually eliminated the need for insulin. That's led us now, in collaboration with Sanford and the FDA, to be in a position to launch a phase 2 trial here in the United States, directly in adolescents. That trial is known as the Sanford Project T-Rex program, and it will be kicked off in January with first patients in. And then finally, as others technologies here, this can be applied across a wide variety of autoimmune diseases, and we'll be looking to start some proof of concept studies with our partners in the near future. The last program is a program in ischemic repair. Here we use CXCR4 and CD34 expressing cells to identify an area of ischemic insult in the body and generate the production of microvasculature to prevent further necrosis of the proximal tissue. We've done a phase 2 trial in acute myocardial infarction patients who had undergone an ST-elevated MI. Those patients actually had their CD34 cells harvested from the bone marrow, but we're moving now on to indications such as CLI and CHF, in which we can harvest the cells directly from the peripheral blood. Makes it less intrusive, less costly, and also avoids some of the limitations on number of cells that are available. Our phase 2 program has already reported results that demonstrated a dose-dependent response on the reduction of major adverse cardiac events. It showed the ability to decrease the size of the infarct, a signal on reducing mortality in these patients, and also a dose-dependent improvement in left ventricular ejection fraction. These are all strong regulatory and clinical indications that promote the advancement of the product into these other indications. Our most advanced program is, in fact, in critical limb ischemia, where we're taking advantage of the new regenerative medicine laws in Japan. We've completed our negotiations with the Japanese PMDA and will be in a position with a partner to initiate clinical trials sometime in early 2016 on that program. We're also working in discussions now with partners to initiate a program in congestive or chronic heart failure that could be done in Japan or actually here in the United States, and we look forward to giving some further information on the progress of that program and its planning in the very near future. Our Japanese program is based on a number of programs that have been done in Japan that demonstrated in CLI the applicability of CD34 cells, and we're looking forward to moving forward in that, as I said, early in 2016. We actually, through our PCT subsidiary, have a small manufacturing footprint in Kobe, and that gives us the ability to actually provide clinical trials in a very efficient and cost-effective manner to support this program. And once again, applicability goes across a variety of other indications, and we're actually in discussions with a number of KOLs here in the United States on programs in stroke, refractory angina, and in Syndrome X. So in summary, I just want to thank you for your attention, remind you that we believe that Caladrius is an exciting and unique company, combining a development pipeline that is spread across a variety of indications, but rooted foundly and focused in autologous cell therapy, and with the added advantage of a PCT subsidiary that not only provides internal expertise and resources, but is a source of revenue and expertise to the cell therapy industry as a whole. I thank you for your time and attention, and I wish you all a great conference. Thank you very much.